Hello, and welcome to the Clinical Care Options Oncology podcast. I'm your host, Brian Topping. Today's episode features expert answers to healthcare professional questions surrounding use of BTK inhibitors for CLL and MCL. This episode is part of a larger educational program titled Addressing Evolving Educational Needs on BTK inhibitor therapies in CLL, SLL, and MCL. During this podcast, Dr. Jeff Sharman from US Oncology and the Willamette Valley Cancer Institute in Eugene, Oregon, Dr. Matthew Davids from the Harvard Medical School and Dana-Farber Cancer Center in Boston, Massachusetts, and Dr. Anthony Mato from the Memorial Sloan Kettering Cancer Center in New York, discuss important topics including how to select between BTK inhibitors and other available therapies for specific patients with CLL and MCL, how BTK inhibitor resistance can occur, and how investigational noncovalent BTK inhibitors might be used in the future. Please visit the show notes for this episode for a link to the complete program, including downloadable slide sets, expert commentaries, and an on-demand webcast from a live CCO event. Now, let's get started and hear what the faculty have to say on these important topics.
The three of us will be talking about a variety of key questions for BTK inhibitors. Here, before we get started, this is sort of a hypothetical patient case. So a patient was diagnosed with CLL and unmutated IGHV. This patient had a 17p deletion and requires treatment. So in your current practice, what treatment would you recommend for this patient? So Matt, in 1 minute or less, do you have any thoughts or reflections on that?
Certainly a patient with deletion 17p should not be getting chemo immunotherapy. I do think continuous BTK inhibitor therapy or venetoclax plus obinutuzumab is reasonable. Say most of us, including myself, for the 17p deleted patients have been preferring the continuous BTK inhibitor strategy just based on longer term follow-up. And, on the other hand, we do see good results with venetoclax plus obinutuzumab. So if this patient did have comorbidities, cardiovascular disease, for example, it's worth considering. But I think I would probably go with acalabrutinib in a patient like this. Yeah, that would be my thought.
All right, we've got these therapies. So sort of patient comes in and has CLL, maybe been following for a length of time. They need therapy. How are you segmenting these patients to determine, who gets what sort of treatment? So how do you shuttle patients back and forth between these options in the frontline setting?
We're left in a situation where we have great drugs. It's a choice between winning options, I would say, and no direct comparative evidence. And so it is a prolonged conversation where we weigh the pluses and minuses for either approach. At the end of the day, if I sit back and I look at the data from your Elevate-TN trial, Jeff, that's the acalabrutinib data, and then I look at the data from CLL14, I feel pretty comfortable that most of these treatments, regardless of which path you take, and get you to the same place with very similar efficacy data and acceptable toxicity data. But it is a comparison weighing risks and benefits and not one based on randomized data.
Fair enough, but I'm going to put you on the spot here, which is, brass tacks, you got a patient in front of you. What are those things that actually do influence your thinking about, a patient may be more appropriate for this approach or that approach? And again, this is more the nuance of practice of medicine rather than comparative efficacy data.
So if they have, active coronary disease, some severe hypertension that's poorly controlled, a bleeding diathesis, they're on warfarin. Just as a couple of things off the top of my mind, it leads me away from BTK inhibitors. If they've had arrhythmia events, it leads me away from BTK inhibitors. If they have underlying renal dysfunction, for example, that would lead me away potentially from venetoclax-based therapy. Most patients are acceptable candidates for either choice, but then you need to take into account the schedule. If somebody is determined to get off of therapy at some time point, then a BTK inhibitor is not an acceptable option in 2022. If somebody is very busy and they can't endure 3 weeks of venetoclax and then 5 weeks of ramp up because of their life, work commitments, personal commitments, whatever, starting a BTK inhibitor is a monotherapy. So it's the comorbidities, it's the schedule. To a certain extent, it may be looking at their genetic profile, take it all together. It's still a long conversation, but I would say most people end up in the middle and either choice are still very reasonable.
So in a patient who's got del 17p, I think it's fair to say we don't have direct head-to-head comparison as you guys have pointed out. And I think there's room for differences of opinion on this one. I don't know that the field has universally coalesced around what the right answer is here. I will tell you, it's my bias to probably start with a BTK in these patients. We know from the CLL14 study, which was obinutuzumab, venetoclax, the del-17p patients, there weren't a whole lot of them in the study, but they definitely kind of dropped out earlier from a progression-free survival perspective than the others. I would be agreeable to treating a del-17p in the context of a clinical trial on obinutuzumab, venetoclax, but curious if either of you guys have a differing take on that.
I personally have no preference. I have no mandate that a patient has to get a continuous BTK inhibitor with a deletion 17p. This is where I might differ a little bit from what Matt was saying. I feel the median PFS, I mean, keep in mind, we're spending a lot of time talking about 17 patients on CLL14. I mean, I wish there had been more patients, but it's such a tiny number to really draw any really strong conclusions about. But at the end of the day, they did have a median PFS of 49 months. And at least from the relapse refractory setting, we do have some ven retreatment data. In my mind, even if you got half of that with retreatment, you're still looking at a median PFS of like 75, 80 months. That's excellent. And I expect that's what you would see with the BTK inhibitor as a continuous therapy. So I think with venetoclax, you need to really think about a PFS too, because I still consider retreatment as the same line of therapy. And I don't think that that's taken into account enough when you think about that choice.
Yeah, I completely agree with that. I will feel much more confident about using venetoclax in that setting once we have at least some retreatment data in the high-risk patients. And I agree with you. I mean, they're likely to respond again. But since we have the data for the BTK inhibitors longer term so far, that's why I've kind of erred on that side. But again, if I had a patient who had AFib or coronary disease who had 17p, I would not hesitate to use venetoclax in that patient.
But for both of you guys, when you're picking, let's just say you've committed to a BTK inhibitor in the frontline setting, how do you pick between ibrutinib and acalabrutinib?
I pick acala. I think the AE profile is better and the activity is the same. And I love the trial that you did that gives contribution of individual components. So I have some data on CD20 that I feel comfortable with. So for me right now, it's a fairly easy decision, although I am mindful of the long-term follow-up data from ibrutinib, the more randomized data for ibrutinib, the overall survival advantages from ibrutinib. But most of the time, for me, AEs trump anything else, and the head-to-head data are quite compelling, and the Elevate TN data are quite compelling from an AE perspective.
So the question was, I read that BTK plus obinutuzumab doesn't work as well in combination. What's your experience? And then the prompt we have is, when do you consider adding anti-CD20 antibodies to BTK inhibitors? So do you want to address that?
Sure, I can take that. So, I mean, first of all, I'm not aware of any data suggesting that the combination does not work as well as single agent BTK inhibitor. I think to me, the question is more about, does the obinutuzumab add anything to BTK inhibitor? We have clear data with rituximab and ibrutinib, both in the frontline and relapse settings, that rituximab does not add to ibrutinib. But I think, the intriguing data set that you've published, Jeff, from Elevate TN does suggest that there may be a benefit of adding obinutuzumab to acalabrutinib compared to acalabrutinib alone. It was a post hoc analysis, but it is, continuing to be present now with 4 years of follow-up. There's sort of, correct me if I'm wrong, I think 9 or 10% or so difference in terms of PFS at 4 years. And interestingly, what you showed was that benefit was not from the high-risk patients. It wasn't the 17p patients. It was actually even the mutated IGHV patients who seemed to benefit the most from this combination. So as I talk to patients, I do mention the combination of acalabrutinib with obinutuzumab. We talk about that versus acalabrutinib. I think, as we've been discussing, there's trade-offs, of course, because with the obinutuzumab, you have the inconvenience of the infusions, some additional toxicities, higher risk of infection, cytopenias. But you have the potential for a longer PFS. And I would say most of my patients, and this kind of segues a little bit into one of the later questions, I think in the COVID era, they've been trying to avoid CD20 antibodies, and so most have chosen acala monotherapy. But it'll be interesting once hopefully COVID is not as active an issue, if some patients will choose the combo.
Yeah. No, I totally agree. I think that the obinutuzumab addition, you get more bang for your buck. You also get a little bit more toxicity. So, I think to some degree it depends upon goals of therapy, and I might favor it more in younger individuals or those for whom I don't anticipate any challenges with anti-CD20 antibodies. Yeah, so younger individuals where toxicity might be less of a concern, because you do see more infections and so forth with the addition of obin. But, interestingly, follow-up, keep an eye on the overall survival difference between those two arms. That's also post-hoc, but there may actually even emerge a survival benefit there. So you talked about COVID-19 and how that's influenced and recommend first-line therapy. I can tell you that for me, it starts with vaccines and boosters. and I've been more recently giving a fair bit of Evusheld to my patients. But aside from those variables, anything else about how COVID-19's influenced how you pick first-line therapy?
I think at this point in the pandemic, and I don't even want to say like recent, but over the last, I would say, several months, I am not really letting the pandemic influence my choice of therapy at this point. If I think a clinical trial is most appropriate, that's the direction I'm going. If I think molecule X plus Y is most appropriate, that's the direction I'm going. So I'm not withholding therapies. I'm not delaying therapies. I'm not giving partial therapies for COVID. Whatever I think is the best CLL directed therapy is what I'm choosing. And then I'm using all those tools you mentioned to try to minimize the risk for patients, regardless of what I choose.
Yeah, so similar approach for us. I mean, certainly early in the pandemic, there were issues around bed availability in the hospital and admitting patients for venetoclax ramp up, et cetera. But that's really not an issue anymore. So the only place is what I mentioned before, where maybe I'm on the fence about adding a CD20 antibody to a BTK inhibitor. So I kind of let the COVID situation kind of dictate that a little bit. But outside of that, nothing else.
So how do you select therapy for patients who've relapsed following either OB-ven, or what about patients who've received a BTK inhibitor? And I guess the relatively obvious answer might be, hey, switch if there's, but retreatment's a different approach here. And Matt, I know retreatment's a topic that's been of interest to you, particularly with OB-ven. The literature is a little bit scant, but do you want to comment on that?
Sure. I mean, I think that is a very appealing option to consider for patients. And, from the relapse setting, we have seen some data that Anthony alluded to earlier that suggests the likelihood that many patients will respond. Small numbers, but it seems like 75% of patients will respond to venetoclax retreatment in the relapse setting. I would imagine that number is likely to be even higher in the frontline setting, but we don't know what that number is yet. So we're in the process of launching a phase 2 trial called the ReVen-G trial, retreatment with Ven-G or obinutuzumab, where patients who have had at least 1 year of remission after their 1 year of ven-obin can get treated with ven-obin again. And so hopefully we'll generate some response data fairly quickly with that study to have a better sense for how effective it is.
Yeah, that's great. And have you guys, zanubrutinib, so sort of an interesting role for that, right? So it is not FDA approved in CLL yet.
So it's true that it does not have a label yet in CLL, but because it's approved in relapsed mantle cell lymphoma, we can, in theory, get access to it off label. It has made its way into NCCN guidelines in CLL. And so I have some limited experience using it in clinical practice sort of outside of the trial setting. It tends to be patients who have had tolerance issues for the other drugs, or, for example, a patient who wants to be on a more specific BTK inhibitor who has to absolutely be on a PPI. As of right now, acalabrutinib does have a drug interaction there, and so zanubrutinib could be a good choice for a patient like that. Although acalabrutinib is now being developed as a maleate formulation that hopefully will come out later this year and we'll get rid of that issue. So for me, it's been pretty limited use of zanubrutinib so far, but it's been a very good experience with the drug. It's been extremely well tolerated, very effective. So I think it'll be great to have it as an option for patients once it's approved in CLL.
Take a step back, Anthony, for me, and maybe if you could talk to me, not just about the fact that it's covalent versus non-covalent, but, if you could talk about maybe like selectivity and pharmacokinetics and how those aspects play into how pirtobrutinib is differentiated from the first and second generation BTK inhibitors.
Sure. So from the perspective of selectivity, this BTK inhibitor is probably the most selective agent that we have available to us. The original preclinical data compared its selectivity versus for BTK versus 300 other kinases, and it was incredibly selective for BTK. If you look at the kinome map, you actually think there's something wrong with it because it essentially only, you only see a dot over BTK. Its PK properties are pretty remarkable as well. They're incredibly predictable, which is what you want to see in a drug. At each individual dose level, the PK properties are essentially superimposable for every patient. So it's got a favorable PK property. It's specific, and that should translate into, a very, very excellent adverse event profile regarding off-target effects, which is what you do see from the clinical data.
No, that's right. And I mean, I think maybe part of what's odd about the question is that pirtobrutinib functions in a way that many drugs work, but not any of the BTK inhibitors work, which is getting into the bloodstream and sticking around for a while. Whereas the covalent inhibitors, are the active substrates are really only present in the blood for a comparatively short period of time. And ibrutinib, perhaps the shortest, acalabrutinib a little bit longer, zanubrutinib perhaps the longest. But it's during that time that you get the covalent modification of the BTK enzyme. And, it's just a sort of different way about thinking about the drugs and the fact that pirtobrutinib goes in and achieves sort of a stable half-life and so forth. Nemtabrutinib is another one that was formerly known as Arcule 531 and is now under development by Merck under the name nemtabrutinib. A little bit further behind, a little bit less data, but I think we'll just have to watch how both these evolve. I do think that pirtobrutinib in this sort of third generation BTK inhibitor class probably has the lead relative to nemtabrutinib from a clinical development perspective. So there is a question in the chat box. Let me see if I can find it again because we got a number of these here. Yeah, so this was also from Barbell, which says, I read that pirtobrutinib can cause mutations. It was suggested to add another drug to avoid this. Which one would you prefer? Let me rephrase the question a little bit to fit with our discussion guide, which is, what are the mechanisms of resistance to covalent inhibitors? And then maybe I'll have Matt tackle that. And then we'll come back to Anthony for some of the newer data regarding non-covalent inhibitors and what happens there. So Matt, do you want to talk about mechanisms of resistance?
Sure. So for covalent BTK inhibitors, this has been pretty well established for several years now that the most common mutation that we see is a BTK C481S mutation. And this was originally described in patients treated with ibrutinib, although it was later seen in patients treated with acalabrutinib. I haven't seen as much data with zanubrutinib there, but that accounts for a good 70-75% of the patients. You also have probably 5 to 10% of patients who have a PLC gamma-2 mutation, some patients who have co-mutation there. So that's accounting for 80-85% of the resistance to covalent BTK inhibitors. And they're pretty widely available in terms of testing now, so we can figure that out as to what the resistance mechanism is for our patients. There's still that small piece of the pie there where we don't know what causes resistance, but that's pretty rare. So that's sort of the general story for covalent. It tends to be in the patients who have deletion 17p or TP53 mutation, especially in the relapsed refractory setting. Although these mutations have been seen in patients progressing after frontline ibrutinib, we don't have that many patients who progress in the frontline setting, so it's not quite as clear as the relapse population.
Yeah. And Anthony, before we get to sort of non-covalent resistant mutations, can you just give a comment about how well pirtobrutinib works against those resistance mutations seen against first and second generation BTK inhibitors?
Oh yeah. So I mean, while ultimately the drug works well in wild type and BTK cis481 mutated patients, I mean the original intent or design of the molecule was to overcome those mutations that Matt already described, particularly in BTK. It works really well. The data that we've presented, and we've all been part of these studies, the three of us, is the data where you can see a high overall response rate for the molecule that's not affected by the presence or absence of a cis481 mutation, for example. And you can see progression-free survival that's not impacted by the presence or absence of a cis481 mutation. So in terms of achieving the goal of addressing the biggest unmet need for a covalent BTK inhibitor-resistant patient, it absolutely does do that. The nice added benefit was that it works great in wild type. It works great in the setting of intolerance. It has a great AE profile. So, but for the unmet need, it really does hit a home run in that department.
And so what are the pivotal studies for pirtobrutinib that will hopefully get a pirtobrutinib label?
Well, there's two, for CLL specifically.
Yeah, for CLL.
There's two relapse refractory studies that are ongoing. One is a time-limited approach, which is venetoclax-based, the Murano regimen, plus or minus pirtobrutinib. And then there is a randomized trial, a 3-arm study, 1-to-1 randomization of pirto mono versus idela rituximab versus bendamustine rituximab that does allow crossover from those control arms back to pirto. So that is another one. And then there's a frontline trial of pirto, correct me if I'm wrong, versus bendamustine rituximab, similar to the Alliance trial. And I've also heard, but I don't know if it's launched yet, of a 1-to-1 randomization versus ibrutinib. I think that's everything, right?
Yeah, I'll tweak one of the ones you said. You called it a 3-arm study in the relapse setting of.
Yeah,
that's actually a 2-arm. Yeah, it's a 2-arm study of investigator's choice. And that's exclusively in patients who had a prior BTK inhibitor. So they have to have had treatment failure from a first or second generation BTK inhibitor. Matt, where do you see these drugs, particularly the noncovalent inhibitors, fitting? Let's just say they were approved today. Where would you use them?
Yeah, I mean, I wish they were approved today because the trial availability has been challenging at times, and I have patients in my practice on a weekly basis who could be candidates, and it's typically those patients who are post covalent BTKI, post venetoclax, where now in the standard setting I might try a PI3 kinase inhibitor, but data from Anthony and others have suggested that the PFS is not going to be great for PI3 kinase inhibitors there. So if I had ibrutinib in that population, I would certainly be using it today. I think it's interesting to imagine sort of in the second line setting, patients progressing after an initial covalent BTK inhibitor, whether you might use ibrutinib before venetoclax, maybe stay in class. Maybe there are reasons why you chose a BTK inhibitor in the first place rather than venetoclax, and those same reasons may still be in place. Maybe the patient has renal dysfunction or difficult for them to do the venetoclax ramp up, et cetera. So it'd be nice to have another BTK inhibitor there. So that's sort of where in the short term where I see a drug like ibrutinib fitting in is sort of post-covalent BTK inhibitors. Now, based on the toxicity profile looking so great, it is definitely intriguing to imagine it moving up into the frontline setting. But I think obviously we would need a lot more data to feel comfortable using it there.
Perfect. Thanks. And actually, I'm going to keep these final questions in mind here, but I'm going to just click through a couple slides because Anthony, this is your data here, just looking at the waterfall plot and then the progression-free survival with ibrutinib, which that's pretty impressive looking. So the one other emerging strategy here, we're just going back to this slide, is the idea of BTK and BCL2 combinations. This is a lot of intellectual appeal to this, which is that you have an all-oral regimen. Maybe there's a unique synergy between the two. And we've got two data sets. One is the GLOW data set. This is a German study, more European study, I should say, than German, which looks at ibrutinib venetoclax versus obinutuzumab chlorambucil. If I'm not mistaken, I think it's the last obinutuzumab chlorambucil control arm study out there. And, no surprise here, you get a significant improvement with the combination therapy. However, I think there's been some question about how well it performed in this setting, which tended to be an older population, sort of had to be above age 65, or you could be less than 65 with certain comorbidities. But, the question to some degree is how much that experimental arm contrasted with either of the BTK monotherapy or obinutuzumab venetoclax. Interestingly, and perhaps in counterpoint, if you look at the Captivate data, which was a US study with a group of patients that were younger. Now, this was not a randomized study, but boy, that looked really quite impressive with the progression-free survival that almost looks like a box top there. So curious for both of your impression when the all-oral regimen is approved, which we're anticipating before too terribly long. Do you think you'll use it?
I can start. I think it's great to have more options for patients, and having a time-limited all-oral novel agent regimen, I think, will be helpful. Based on what you were alluding to, the differences between these two studies, to me, Captivate and GLOW, were more around toxicities rather than efficacy. I think the efficacy looks great to me in both. But in those older patients in the GLOW study, there was more atrial fibrillation, cytopenias, and even some more serious cardiovascular events that were a bit concerning, and that wasn't really seen much in Captivate, where the regimen was much better tolerated. So for me, the patient would be kind of that younger, fit patient, wants a time-limited therapy, doesn't like the idea of being in the infusion center all day for 6 months with the obinutuzumab. So yeah, I do think it'll have some use, but as we've alluded to in other parts of the conversation, we're all using acalabrutinib more. So I would be actually much more intrigued by the possibility of acalabrutinib with venetoclax, which we will not have as a labeled regimen that soon, but hopefully at some point in the future.
Yeah, great. Anthony, do you have any comments on where you might use that?
I agree with what Matt said. I think it's going to be it'll probably be a small minority of patients. They'll be younger, they'll be fit with a poor risk feature, and they will be interested in something time limited. I'm a little disappointed with the GLOW data set in terms of first of all, the PFS, and then more importantly, the AE profile. So for my average CLL patient who's in their 70 plus years being treated, for me, CLL 14 remains a standard of care for a time-limited approach.
I'll address some of the topics in mantle cell lymphoma, another place where BTK inhibition is really important. For patients who experience relapse, 3 different BTK inhibitors have been approved: acalabrutinib, ibrutinib, zanubrutinib. All of them were approved on the basis of single-arm phase 2 studies involving about 100 to 140 patients. And all of them looked relatively similar, allowing for all the typical cross-trial comparison analyses. Frankly, if you're going to pick between BTK inhibitors, I think that the CLL literature, which included a number of head-to-head studies, might be more informative than any of the unique studies in mantle cell lymphoma. There is a trial right now of pirtobrutinib, which compares pirtobrutinib to investigator's choice of acalabrutinib or zanubrutinib. So it'll be interesting to see how that reads out with time. And in terms of BTK inhibitors in the first-line setting, very limited data for monotherapy, mostly kind of retrospective anecdotal data in individuals who are older or who may not tolerate more aggressive chemotherapy. There's some question about using BTK inhibitors in those mantle cell lymphoma patients who have either 17p deletions or TP53 mutations, because they're not going to do particularly well on chemotherapy. And I think that time will tell where the noncovalent inhibitors may fit in these settings, particularly now that we have a head-to-head study comparing the non-covalent with the covalent inhibitors. So what are the key safety concerns for BTK inhibitors? You kind of alluded to it when we started talking about how you segment these and so forth, but did you want to sort of address this question?
Sure. I think while there's a lot of off-target effects, if you're talking about key safety concerns, I would say cardiovascular toxicities, arrhythmia would be number one, either atrial or ventricular, hypertension, and then the downstream effects of either or congestive heart failure or coronary artery disease, peripheral vascular events, or stroke. Second on the list would be bleeding events. And for me, third on the list would be infections. And I think those are probably the most concerning risks associated with BTK inhibitors. I'm not sure if either of you guys would add anything more to the list.
That really fits with my experience. Matt, did you have anything you wanted to add?
No, I totally agree. I mean, to get to the arthralgia, myalgia point, that is quite common, but I've found it usually be manageable, usually with supportive care, dose adjustments, et cetera. Sometimes I have had to change to a different BTK inhibitor, but that would be, for me, often a reason I'd want to stay in the class to try to find a BTK inhibitor that's tolerated. But some of the more salient events that Anthony mentioned, like severe bleeds or cardiovascular issues, I've sometimes had to switch patients to venetoclax, for example.
Thank you very much, Dr. Sharman, Dr. Davids, and Dr. Mato. And thanks to you, the listeners, for joining us. As a reminder, to view the full program addressing evolving educational needs on BTK inhibitor therapies in CLL, SLL, and MCL, and to download slide sets associated with this discussion from the Clinical Care Options website, please click on the links in the show notes. As always, thanks for listening.